But what I intend to present here today is just some summary of some data I got in this most recent work carried out only in the Amazon. And as the author said, in 2005, I was the science director of Butantan, and Butantan was planning an extension of their activities to the Brazilian Amazon, more specifically to Santarém, here in the west of Pará State. And I was responsible for the interinstitutional contacts, projects, and trying to organize the university and research institutes in the region. So for doing that, the research project that I suggested to start these collaborations was regarding what I knew a little bit how to do, what was understanding composition and variability of Botrops atrops venom, which is the most important snake concerning snake bite in the Amazonian region. And then trying to correlate this venom composition with human symptoms of snake bite, getting close to the clinics of and the physiopathology of the accidents, and also to do some preclinical tests for the efficacy of Butantan antivenom in the region. As some of you may know, there is an important issue about Butantan antivenom, which is in Brazil, we have a standard immunization tool for antivenom production. And Botrops atrops venom is not included in the pool. So it was very important to assure that the antivenom of Butantan was efficient in the region, and it was a very interesting thematic to work there. So we started here, and with the increase of the collaborations in the Amazon, we could have some samples from other parts covering quite a nice geographical area. So the first approach was trying to correlate. We knew the first approach, no, one of the points we focused was trying to understand the influence of snake population genetics in the venom composition. We know there's a lot of problems in the classification of Botrops atrops. Some authors suggest different populations, even different subspecies. And then for that, we did some genetic tests on snakes in the west of Pará State here, from snakes from the north, here in Oriximiná, collected in a pasture area, comparing to snakes from the south of the Amazon River, collected at Flona Tapajós in Santarém area, and also some areas in between. This savanna area, which was just in the shore of the Amazon, south shore of the Amazon River, and the Várzea, which is the floodplain area close to the north shore of the Amazon River, but in the middle of the river, actually. And in these genetic studies, we could detect two different populations based on rat tags, one in the south, one in the north. And the Varzian savanna already showed some mixing of the genetic markers of both populations. So, going to the venom composition, we did individual proteomics and transcriptomics of 5 individuals, 2 from the north, 3 from the south of the Amazon River. And we see here the almost similar venom composition, with the major toxins here represented in blue being the snake venom metalloproteinase, in all of them, followed by C-type lectin-like toxins in orange. So the venoms were similar. However, when we tried to analyze the expression of each isoform, we detected that venom from the north uses different isoforms of toxin families than venoms from the south. Although the protein families were the same, the isoforms were different. However, the venom biological activity, like the coagulant, minimal coagulant doses, hemorrhagic, and also the lethal activity, were very comparable in south and north collected states. However, in the Varzea, we saw some difference regarding hemorrhagic and coagulant activity. So the other thing that we looked for, if there were any influence of the snake habitat for the snake venom composition. And then we compared the snakes from the north, pasture, floodplain, Varzea, and here the forest, already said before, with these snakes from this degraded area close to this forest area, just to compare with the pasture snakes from the north. And here, analyzing the expression of the isoforms, again, we see that a different set of isoforms, the clusters here separately, were present in the venoms from Oriximiná, the pasture. And again, probably related to the genetic difference in the snakes. However, even here, the most predominant snake isoforms were metalloproteinase, similarly to the snakes from the forest, which were similar to and closer to the snakes from the degraded area from the south. However, we saw marked difference in the isoforms, making this variability in the venom of floodplain snakes, and here as well with this particular cluster here. And what is interesting here is that it included mostly serine proteins. As consequence, the venoms from the Varzea floodplain showed more stronger procoagulant activity, as small the graphic, higher the activity, and smaller hemorrhagic activity. The LD50 was also a little bit lower in the Varzea snake venoms. However, it was not statistically significant. And the other question that we did concerning venom composition was if there were any individual variability in snakes collected at the same spot. So here we collected a few snakes from the forest, the pasture, the same areas as before, degraded and Varzea. And again, we can see here by chromatographic profile in reverse phase HbA1c that the most predominant in all of them is the snake venom metalloproteinase. However, this region is quite variable among the venoms, and this region usually includes the serine proteinase, phospholipase, and also C-type lectin-like proteins. So what is much interesting about this study was that the 4 venoms from each area were tested individually for their biological activities. And we see strong variation on hemorrhagic and myotoxic activities among these venoms from the same spots. Note that the average is similar, but there is a very important difference among the venoms. Concerning coagulant activity, they were much more similar, except some of the venoms from Varzea. What is very, very important here was that when we tested the time each of these individual venom takes to kill mice or birds, we see that some venoms, like this one here, kills mice very quickly and slowly for birds. And here as well, like this one, very quickly here, and almost most of the birds were not killed. On the other hand, some venoms from Varsia killed birds much quicker than most of them, very, very quick, than they killed mice. And it was probably related to some advantage for the Varsia snakes to survive in extreme conditions of this habitat that keeps flooded for almost half of the year. This geographical difference these differences in biological activities were also observed when we compared the geographical variability of Botrytis venom. And here, we tested Presidente Figueiredo snakes, venoms from Presidente Figueiredo snakes, which is in the Amazon state, and venoms from São Bento, which is in Maranhão state. So two very different areas in the Amazon, comparable to the Belterra venom, which is our standard. What we can see is that, again, all show the metalloproteinase as the most important or most abundant component, variability in this region of the chromatogram, and it's very clear the absence of this peak in Presidente Figueiredo venom, which is the major serine proteinase of the Belterra venom. And in São Bento, Maranhão, the venoms did not present phospholipids like the K49 phospholipids. And also they don't have this peak, which corresponds to a P1 STMP that I will talk in the next slide. So, as a consequence of this difference in venom composition, São Bento snakes had a venom with a more strong thrombolytic activity, the smaller, the more strong, more hemorrhagic, but less dermonecrotic. In the position, the Presidente Figueiredo venom was recognized by their low lethality in spite of their high dermonecrotic activity. Somehow, as if the lethality
sorry, Anna, is to have 3 minutes, sorry.
Okay, in the direction of São Bento and decrease in the dermonecrosis in Presidente Figueiredo. So we did the characterization of relevant variable isoforms, and just to make a long story short, we characterized the P1 class as PLP, very strong hemorrhagic and dermonecrotic. It was a characteristic of the tree. And we also characterized a procoagulant, prothrombin activator, and factor 10 activator in vasovagus. We did some correlation between the isoforms present in snakes brought to the hospital from 14 patients. So the venom composition of these snakes was correlated to the symptoms of the patients. And then we confirmed the relevance of metalloproteinase and serine proteinase for bleeding, pain, edema. But also we showed the importance of CTLs and other isoforms for bleeding and blistering. Concerning the antivenoms, the butantan antivenom reacted in ELISA with Botrytis venom and neutralized hemorrhagic lethality, coagulant, and dermonecrotic activity of most of the samples tested. We had some problems in neutralization only of the coagulant activity of the Varsia venom, and it was mostly due to lack of neutralization of factor 10 activator present in this venom. However, if we adjusted the dose of the antivenom, the neutralization was perfectly achieved. So just to finish, this data was included in a series of publications, always together with people from the Amazon, maybe that's why we managed to make this big network. But also it gave us insights to analyze lots of data in the clinical studies carried out in the region. So most of the new insights in Quadriceps snake venom and envenoming, we did a lot of work in training people in the Amazon. And these numbers are quite impressive. And we are very proud to collaborate today with this group from Manaus. And I want to say here that when we started the work, it was a very famous group for clinical studies on malaria. And we were able to envenom them. And they are now one of the most productive research groups studying clinical aspects of snake bite in the world. And I thank to welcome Monteiro the possibility to work with them. So just to finish, I think my principal achievements in the working in the Amazon was the training of professionals from the Amazon to act in the control of snake bite in the Amazon. So thank you very much for your attention, and again for awarding me with this lifetime achievement in toxicology 2021. Thank you very, very much.